CBSE Biology, Class 12, Chapter 5, Principles of Inheritance. Let us learn the biology chapters through questions and answers, which most of the students prefer. Note that PG here is for page, PR for paragraph in your textbook. Questions are in yellow color while answers are white. How Morgan came to know that two genes sometimes do not segregate independently of each other. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex-linked. The crosses were similar to the dihybrid crosses carried out by Mendel in pea plants. For example, Morgan hybridized yellow-bodied, white-eyed females to brown-bodied, red-eyed males, and intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other and the F2 ratio deviated very significantly from the 9:3, 3:1 ratio expected when the two genes are independent. What is linkage? How Morgan coined this term? Morgan and his group knew that the genes were located on the X chromosome and saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combinations were much higher than the non-parental type. Morgan attributed this due to the physical association or linkage of the two genes and coined the term linkage. Morgan coined the term linkage to describe the physical association of the genes present on same chromosome. How Morgan described the term recombination? Recombination is the generation of non-parental gene combinations. Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were very tightly linked, showed very low recombination, while others were loosely linked, showed higher recombination. How the two terms linkage and recombination are interconnected. Morgan and his group found in Drosophila that the genes white and yellow were very tightly linked and showed only 1.3% recombination, while white and miniature wing, loosely linked, showed 37.2% recombination. How the work of one of the students of Morgan helped in Human Genome Project? Morgan's student Alfred Sturtevant used the frequency of recombination between gene pairs on the same chromosome as a measure of the distance between genes and mapped their position on the chromosome. Today, genetic maps are extensively used as a starting point in the sequencing of whole genomes, as it was done in the case of the Human Genome Sequencing Project also. State the strength of linkage between two genes. Here are the results of two dihybrid crosses conducted by Morgan. Cross A shows crossing between gene Y and W. Cross B shows crossing between genes W and M. Here dominant wild-type alleles are represented with plus sign in superscript. Note that the strength of linkage between Y and W is higher, because they're closer than W and M.
End of this part. Dear students, share your doubts and questions to this email given below. The question answers of this chapter will be continued in the next video of CBSE Biology Class 12.